Okay, thank you for sticking to the end of the meeting, and I'm going to tell you about our thoughts developed over a number of years on the idea of plasticity and how it affects various aspects of tumor progression. I want to start with a very brief sort of background of where we came from, and originally when we started to think about cancer, which was already about 15 years ago, we decided that the most interesting question would be to try to understand a little bit better how cell fates, such as were indicated by this EMT transition, would give rise to metastasis. So our first efforts were to think about metastasis, and this was a standard cartoon developed, in this case by Bob Weinberg, about the thinking of the cancer biology community during that era, 2013. And the idea was very simple. The idea was EMT was the answer to the question of how is it possible for cells to pick up, move from the primary tumor, make it through the various stages that it has to in order to establish a secondary tumor in some metastatic site. EMT is the epithelial mesenchymal transition. We just heard about it, so I don't need to go through it in great detail. Borrowed from the developmental biology community, where it was a well-established way in which cells could change their phenotype and give rise to development of various organs. And it served the purpose of trying to explain how it could work. Now, in this picture, you see that there's primary tumor cells. Those are the green things. You see there's metastatic tumor cells. Those are, again, green in a different organ. And then you see cells going through some transition states, going from epithelial-like to mesenchymal states. And of course, nothing happens instantaneously. So the idea that there would be a transition from purely epithelial to purely mesenchymal states, the states would then leave the primary tumor individually and go and create the metastatic lesion. So the notion was there were things called partial EMT states, but those were all transitional states. Now, several of us, and Jinhua and our group sort of in parallel, looked at this question from a more systems biology perspective, trying to understand what was the underlying biology that was giving rise to EMT. This is just an example of that work. These models all in that era were relatively small, not really being driven by single-cell RNA-seq data, because frankly, there was no single-cell RNA-seq data in 2013 and 2014. So being driven by bulk data, being driven by various biology experiments that indicated which were the leading factors that seemed to be contributing. Here is an example of what a model looked like from that era, a medium-sized model. There are different factors. Some of these are transcription factors. Some of these are microRNAs. There's effectively what Mohit and Kishore described as eventually as a team-like structure, where sort of a lot of the factors sort of collaborate with each other, such that you get one team that's sort of pushing for transition from E to M, and another team of factors that are pushing for retaining the epithelial state. And you can study this model in detail and try to understand what are the states that are allowed by the topology of this network. And I'm going to skip all equations and all details of calculations, but this was our study of this in that era. And if you look at the right, this is an attempt to use this model to produce what you might expect to see in some population of cells. Think about a cancer cell line, and you have some population of cells in that cancer cell line. They're heterogeneous. They have different values of parameters, but they all have some topology of this network that's conserved because they're all supposed to be isogenic. So therefore, if you look at that graph and you don't worry about all the labels or whatever, what you see is that well, there are states that look like epithelial states, where all the factors that you would have naively associated with the epithelial fate are high, and all the factors that you would associate with the mesenchymal state are low. And you see a very consistent pattern. The top 40% of that effectively single cell picture, which of course we couldn't compare directly to experiments at that point. The bottom are sort of the inverse of that. You see mesenchymal states, and those states are all the mesenchymal factors are high, all the epithelial factors are low. They're very well established, very well differentiated in this model. And then the surprise was, and I said this was shown in a variety of models by a variety of groups, was that there were other possibilities for these networks with this topology. The other possibility are things that we called hybrid, EMT cell states. They're cell states where you're expressing some mixture of epithelial and mesenchymal properties in the same cell, not at the population level. The same cell is expressing both populations. And the thing to compare this statement to the previous cartoon is these are not transition states. Repeat after me. These are not meant to be transition state. It's not just states that you get when you go from E to M. They're actually stable at some level. I mean, they can interconvert at some level, but if you study a population, some percentage of your population can be E/M, hybrid, partial EMT, whatever you want to call it. Now, the other feature is that these cell states are somewhat different than the E and M states, and you can even see this in this picture. But we study this in much more detail, which is that these cell states are much more vague. They're not all the same. You have a variety of possibilities. Their transcription pattern is very fluid. It's not very fixed. And that has the feature that you might imagine that you can transition among these different states if you need to, if some of them are better adapted to the environment than the others. You have a question?
Yeah, I just wanted to understand. So is the thing on the right basically kind of simulated from the thing on the
Yes.
left? Okay.
Yes.
Great.
It's simulated by a strategy of trying to create a population by having some controlled variation of the parameters in the model described by that topology of
To
that.
get these mixed states
Yes, right.
stable
Yes.
as part of it. Okay.
Okay. Yes. I was afraid I'd get stuck here, but go ahead. That's okay. No problem.
You interrupt anyway. Understand why people emphasize that the hybrid state is not a transition state.
Because
a flat attractor, is that a transition state for you or not?
It's not a transition state.
Yeah, it's not.
It's not.
When is something a transition? Because a flat attractor.
A transition state means to me two things.
Yeah.
First of all, it means that to go from E to M, you must go through this state.
Yes.
That is not true.
Okay.
We showed actually in Katie's talk, you showed that you can have direct transitions between different phenotypes. In her case, it was fibroblasts to neurons. People have shown that you can have direct transitions from E to M without going through one of these intermediate states. So in that sense, it's not a necessary transition state between them. And also, when you say the word transition state, you imply it's much more transient. These don't have to be transient. In the first experiments that we did looking for these things, we could repeatedly culture lung cancer cell line, one particular one that we worked on, where you saw these cells there for a month or longer, staying stable.
Herbie, for the first point, though, I have to push back because I don't think Katie showed that there was no intermediate state which belonged to the two endpoints.
Yeah. Yes, that's true.
Right?
Yeah.
So we don't know it.
No, I didn't say that she showed the no transition state. I'm just saying that that's true. Okay.
Yeah.
But that's a long discussion about Katie's paper, which we don't want to. So actually, I asked her about that because if the transition state that you don't see in that work is the induced pluripotent stem cell state. You don't see that you're going up to a stem-like state and then going back down. She does not see that. There can certainly be states that you can call transition states in the pathway. That we don't know.
Like I guess what you could think of is if they're two completely different cell types, the question is, do you go through a stem cell-like state?
Yeah. So she has. So there are. I don't know her experiments. I asked her that directly during her talk, by the way. But in other versions of that same system, going from fibroblasts to neurons, it's been shown very definitively that you do not go through stem-like states. Yes.
In this case, are these stable? Or sorry, I didn't catch you.
Yes, these are stable. I mean, there's interconversion at some rate, but that's just always okay. Now, so I'm going to skip, 8 years worth of work by numbers of people and numbers of labs and numbers of computations and try to explain to you why this turned out to be important. And, who cares if there's, you call your states E, you call your states M, you have hybrid states, who cares? So the answer, I think, was, the first paper that really, I think, established this, got over the barrier to being accepted in the cancer community was this experimental paper by a European group where they asked the question in a mouse model of was there something special about those cell states versus the normal E and M states? And so what they did is they grew a mouse tumor. This is genetically engineered mouse tumor. The details you can look up. And they then extracted the tumor. They then sorted the cells based on surface markers using, at that point, relatively, not completely established, but some guess as to what would be good correlates of these different states in those particular markers they had access to. And then they re-injected back into new mice, and they asked which of those populations were much more capable of generating new tumors and new metastatic growths in those new mice. And the result, summarized by the News and Views article in this paper in Nature, was the following statement, that they observed that if you did that sorting, that the cells that were intermediate, the ones that exhibited properties that could be identified with intermediate cell types, were the ones that actually were able to do tumor initiation much more effectively than the others. This was established in vitro before this, looking at just sphere assays and other in vitro organoid formation, but this was sort of the first direct in vivo version of showing that these cells were in an in vivo context the ones that could be much more effective at generating new tumors. I always, and this in some sense, it's a shame that Mohit is not here, because we just heard about Mohit from the previous talk, and I'm going to also cite Mohit's paper when he was my student, and I always like this statement that appeared in this paper, because luckily, I mean, one of the sociological problems that has to be solved is getting cancer biologists to believe people doing modeling. And by modeling, I don't mean data analysis. I really mean modeling. And luckily for us, this group actually did quote our paper. They actually did understand that people in the systems biology community, and Jinhua and myself, people who are working on this, really did predict something like this. And so on the one hand, they say it was very surprising to them, which they probably had to say to get it published in Nature. But on the other hand, they did cite our paper, which actually predicted this. So I think this was really a pretty good thing. There's more papers after that. I wanted to just mention this other paper because I love the technology in this paper. We know we've heard a lot about lineage tracing and whatever, but this paper uses a technology which is I don't know of any really other paper in the cancer community that's doing this, where the lineage tracing is actually something that you can go in and change dynamically through a CRISPR system. So you actually inject into the cells not a fixed barcode, but an evolving barcode. And then you can go and do a phylogenetic reconstruction, and you can trace not only the original cells that whatever you see came from, but also what some types of transitions were the cells along the way by tracking these evolving barcodes. So I think this is a very nice technology.
Herbie wants to give us some history of it.
Yeah.
The reason I have this one is that before this, right? Several years ago, there are two back-to-back Nature papers.
Yes.
They use lineage tracing, claim that hybrid stage does not contribute much. Ever agree that this showed it, right?
And then there were papers afterwards that showed that they were not. Yeah. Anyway.
Look at the right thing.
Yeah. Anyway, so this paper also, we like to quote it because first of all, I love the technology in this paper, but also they also came to the same conclusion that when they tried to back up and do this phylogenetic reconstruction, they also decided that the cells that were giving rise to the metastasis, this is in a pancreatic cancer model, were again these hybrid cell types. Okay, so this is just a summary from the biology community a couple of years ago of now I would say the current picture of now you have a sort of continuum of spectrum of EMT cell states. And the ones in the middle have certain unique properties. They have certain unique biophysical properties. But from the point of view of what I was interested in, what continue to be interested, they're the ones that have the best adaptability and also the most likely are contributing to metastatic. Now, you can then ask the question, especially if you remember the previous talk, you have this whole tumor and you have various cells undergoing EMT, and you can ask, okay, what decides if a cell is going to undergo a full EMT or if it's only going to go to one of these partial states? Remember, this is not a transition state. It's an alternative final state for some of the cancer cells. Now, we could address this question in a number of ways. One was just a reanalysis of some published single-cell RNA-seq data. We're now up to the 2024 era, so now we did have single-cell RNA-seq data. This was originally the data set was done. We collaborated with a group of Mani to generate this data set, and then this is a reanalysis of that data set. And in this data set, what you saw is you took a cancer cell line, breast cancer cell line, you subjected it to a standard drug induction of EMT via giving TGF-beta. This is MCF10A, for those of you who want to know what line it is. And it turns out that when you look at the trajectories in the single-cell RNA, you see that the cells undergo 3 possible different trajectories based on the original analysis. Some cells undergo full EMT. Some cells do nothing. They sort of just sit there and ignore your signal. And the others undergo something that we could identify based on markers and based on stem-like properties that looked like they had undergone partial EMT. And now the advantage is you can go backwards using computational methods, which are not perfect, but are better than nothing, in particular optimal transport, which allows you to connect cells at one time slice compared to another time slice. We had 5 time slices for this data set, so we could go back and ask, what were the properties of the cells at the initial time before they were given the stimulus that seemed to bias them into one of these 3 possible cell fates, either doing nothing, undergoing full EMT, or undergoing this partial EMT transition. And the answer in this system was that the most likely indicators of what the cells would do were the expression levels of various epigenetic related enzymes. So this is a hint. It's not a detailed analysis, but it's a hint that you should begin thinking not just of the transcription factors, but of the underlying chromatin epigenetic related regulation of accessibility of DNA, all that other stuff that the systems biology community has been slowly trying to come to grips with. Now, just to remind you, we heard a little bit about these talks. The basic part I'm talking about is you have this wrapping of DNA into nucleosomes, and those nucleosomes, and in particular some of the post-translational modifications of those nucleosomes and also of the DNA itself, affects the accessibility to transcription factors of that region. It's another layer of regulation sort of pasted below the transcriptional regulation level. So the first experiment, that I described, the reanalysis, was nice because it gave us some indication. But this experiment really helped us try to understand a little bit better what's at stake when you try to think about this problem. This is an experiment from a postdoc in Bob Weinberg's lab, Yun Zhang, who now has his own lab in Beijing. And they, again, took the same strategy. They looked at a cell line, not exactly the same one that we had done. This is HMLER, for those of you who know breast cancer cells. And they, again, did the same thing. They looked at a drug induction of EMT, and they found that in their particular experiment with their concentrations, cells underwent sort of either did nothing or they underwent EMT. And what they were interested in is the cells that did nothing. And what they did is they did a CRISPR screen. I guess in these days I have to tell you what CRISPR screen, CRISPR knockout screen of what could rescue the cells that were unable to undergo EMT. And the answer, again, that they found, and this is in the title of their paper and published a few years ago in Nature Cell Biology, was, again, they found things that modified the epigenetic marks of the various factors that in this particular cell line were the drivers of EMT, things like snail and Zeb and things like that, which we're not going to discuss in detail. So there were many interesting features of this data that they showed. First of all, they again showed that one needed to begin to think about these epigenetic regulators in the context of EMT. They also found some very surprising results. For example, one of their surprising results is that these were rescues of EMT. Now, if you're rescuing EMT, you would think, well, a natural thing that would rescue EMT is suppressing an epigenetic writer of an inhibitory mark on the promoters of EMT transcription factors, right? That's what you'd expect. So they found that. So PRC2, for those of you who are not into epigenetics, is something that puts methyl groups on the 27th lysine of histone number 3. And you have to learn all this stuff if you're going to talk about this, which is really annoying for those of us who are not biologists. This is a repressive mark. It was shown directly in this system that repressive mark could sit on promoters of various EMT factors. Mostly, in this context, Zeb was the primary factor which was governing the EMT transition in this system. And excuse me. And so if you knock out PRC2, all of a sudden you've removed a repressive mark from a leading EMT transcription factor, and therefore you would expect that would allow it to be more easily induced to undergo EMT. More surprisingly, if you knock out a different epigenetic factor, KMT2D, this is a methyltransferase. Even though it's a methyltransferase, it's putting methyls in a different part of lysine. This is lysine 4 for histone 3. This is H3K4 mark. And this methylation mark is actually an activating mark of, and these, the fact of which are activating and which are inhibiting for promoters is a well-known subject matter. So if you knock out the activating mark, you also get now induction of EMT that you didn't get before. So for those of you who are interested in the gory details of how you can make a model which incorporates that, you can read our paper from a couple of years ago done by Molly, who is now actually working for Revolution Science, as we discussed in great detail, who is a great guy, he was a postdoc at Northeastern at that time. And the secret, I'm not going to tell you all the details of the model. First of all, so we do couple directly these epigenetic mark creators and destroyers to a genetic network, leaving out all the details. This is I hope to be a growth industry in the next few years of people doing modeling, begin to think about these epigenetic layers. And how am I done? I'm done with time? Okay, I'll be done I figured I'd get through 2 parts and not the last part. So the answer is that, when you write down epigenetic, it's sort of like doing CRISPR. You have to worry about off-target effects. When you knock out epigenetic marks or when you add epigenetic marks, you're not just adding it to your own promoter that you care about, you're adding them globally in the cell, and you have to take that into account in your model in some way. So that's the details here. Okay, so I'll quit that. Currently, we're trying to do similar things. Progression for tumors is not just metastasis, it's also drug resistant. We've been looking at drug resistance, and I just want to refer to some experimental paper that is that I should say affected our thinking, but actually it affected our thinking in the sense that we had a model that could at least try to explain this, showing that, again, when you think about ability to respond to drugs, you should not think of them as binary choices. You should not think of them as being a resistance phenotype and a non-resistant sensitive phenotype. There's a continuum. And what's the continuum dependent on? It's probably dependent on the chromatin structure of how easy is it for you to adapt to the drug once you're given the drug. And if you believe in that concept, you can make a model which tries to take that continuum nature into account. And this is an experiment, I think maybe one of the first ones that sort of directly shows that continuum. This is an experiment from Itai Yanai's group at NYU showing a continuum of drug response phenotypes that they can access to in again, an in vitro experiment. And those of you interested in that, we have a paper under review which tries to describe how you can begin to think of drug resistance also as having a range of possibilities, not just a binary transition. Okay, so we're done. I've talked about intermediate states and plasticity. Much more work needs to be done. And if this works, I wanted to leave you with one cultural reference. Anyway, it's July 4th tomorrow, so I have to give you an American cultural reference. Well, from my childhood, there was a very famous movie that had a lot of effect on those of us growing up in the 60s. And I'll show you a clip from it if it works. Let's see, is the sound going to work?
Just one word. Just one word.
Yes, sir. Yes,
Are you listening?
Yes, sir. Yeah.
Are you listening?
Yes, sir. Yeah.
Plastics. Plastics.
You got the picture. Okay, so the idea is, I don't know how to go back, but that's okay. This is, so I just have one word for you, plasticity. Okay, thank you very much.
In the interest of time, we'll move on.
Okay, we can find me afterwards. That's fine.
Russell? I guess if anyone has any questions for Herbie while Russell's getting set up.
Yeah, you had those
please go ahead.
questions over there. Yeah, go ahead.
I was just wondering, incorporating the epigenetic information into the model, are you essentially using it as a binary variable, or is there any continuous information?
Well, we're doing it as a sort of binding and unbinding effect. So in other words, we allow for binding of these enzymes to various promoter regions, and then when it binds, it has some rate at which it writes that mark. And so the mark itself is a binary variable, right? There's either an H3K4 ME3 mark or not, but the dynamics leading to that are binding and unbinding of these enzymes, which are treated as continuous concentrations of enzymes. And then,
sorry. So to what extent do you think that the epigenetic information, like given if things are really in some sort of steady state, the epigenetic information is contained within the mRNA information? Like,
in other words, do you have to measure it separately?
Yeah.
That is a very good question. I don't know the answer to. I mean, people are measuring it separately, at least to some extent, with ATAC-seq. So I certainly think that that's a good idea. In other words, since we know that that's a relevant aspect of the transcriptional program, if you have, the single cell, the same cell RNA-seq and ATAC-seq, it seems crazy not to be incorporating that information in whatever algorithm you're developing. So, but your question is, I think I don't know the answer to, is the information in the transcriptomics itself enough represented of that you could use that to get a good model? So there are clearly problems with that from an absolute standpoint, because, for example, if you believe that writing and reading chromatin marks is an important indicator of how things are going to evolve in the future, then at the very least, if you're just looking at the transcriptional thing, you do not have a simple Markov, ODE. It's not just this current state of the system that gives you the evolution. There's some factors from the previous. It takes a while to write these factors, and that's a history-dependent effect. You completely from the transcriptomics perspective. So if you can take that into account in some way, then probably get by with just transcription. But it does complicate your life if you have directly the epigenetic information, and I certainly think you should be using it.
Thank you very much.